Welcome back. We have in the last session we have given you some basic concept about the structure of nucleic acids, mainly the deoxyribonucleic acid, the DNA. We know that these two scientists which are shown here, their pictures, Francis Crick and Watson, they proposed this double helical model. The most beautiful aspect of this double helical model is that the hydrogen bond interaction between the bases. So, the bases are stacked inside, again I said, and G forms hydrogen bond with C, and the number of hydrogen bonds between GC are 3 and number of hydrogens between A and T are 2. So, whenever one strand has a A, so in the opposite strand, in the other complementary strand, a base will be right there, which will be A. If it is T here, then that will be A. So, if it is C here, that will be G there. So, all complementary bases will be lined up, and then they will pair with the bases of the primary strand. Okay. Now, while doing that, they adopt a helical conformation, and that is why it is called double helix. Now, the double helix, the structure is shown here, but I have made a kind of a model to make it a little bit clear, to show you a double helix which is made up of just aluminum foil. You can see that. Okay. This is a small piece of the double helix. Okay. So, double helix, you have to notice something. First of all, these two helices are right-handed helix. Again, I told you about in case of protein, what is the right-handed helix? That means you have a right-handed screw, the way it goes, that is called right-handed. So, there are two strands. Interestingly, both the strands are right-handed. Okay. And so, what is meant by the anti-parallel strand? That means on the top, if this strand is 5 prime, that means the other strand, if this is the end point, that will be 3 prime. Okay. And on the bottom, exactly the reverse will happen. Okay. Now, some of the important features are that what is the pitch of this double helix? What is the pitch of this helix? Pitch means, again, pitch means if you take one strand and make a complete rotation. If you traverse from one point and go to a complementary point in the same strand, then the rise in the height, that is what is called the pitch of the helix. Okay. So, here the pitch of the helix is basically, you have to take, I just show you, suppose I take this point, and so the pitch will be from this point, take the same strand and go below and take the same other point. So, the distance between these two will determine the pitch of the screw, the pitch of the helix, okay, in this case. So, if I now go to the diagram, so diagram means what is the, so this is your one strand that is going like this, and this is the other strand that is going like this. So, what will be the pitch here now? The pitch will be, if you take this red strand here, the pitch will be follow this red strand and go to a similar point. Similar point means this one. So, this distance is the basically the pitch, but it is said that what is the this is called, if you take a complete turn, then what is the, for one complete turn, what is the rise in height? Okay. Or what is the, if you start from here, what is the lowering of the height? If you start from here going to there, rise in height. if you come from here to there, that is lowering of height. Okay. So, the pitch of this is what is complete turn. So, this complete turn in case of the DNA that was used by Watson and Crick, that was by the way called B DNA. There are again different kinds of DNA. One is B DNA, another is A DNA. We can come to that one, but mainly the DNA that we have is mainly are all, and another is Z DNA. But the DNA that Watson Crick used was B DNA, and this is the most common DNA that is encountered. Okay. So, we will first concentrate on the B DNA. In the B DNA, this one complete turn or the pitch is 34 angstrom. So, that means one you go from here to there, you cross 34 angstrom in length. Okay. And then for each, from one base pair, if you go to the next base pair, that is actually 0.34 angstrom, 0.34 angstrom. Okay. So, if that be the case, that means if I ask that how many base pairs are there in one complete turn. So, that will be 34, sorry, this is I am sorry, this is 3.4, not 0.34, this is 3.4 angstrom. Okay. And this is 34 angstrom. So, number of base pairs in one complete turn will be 34 divided by 3.4. So, it is approximately 10. So, there are 10 base pairs in one complete turn. In this A DNA, there are some DNAs which are more compact than this. There may be around 11 base pairs, that is called A DNA. Okay. But as I said, the most common DNA is the B DNA, and Z DNA is little different. That is, that consists of the very unusual form of DNA which are left-handed double helix. A DNA is the one which is more compact, and this compactness comes from when they are, remember this, the DNA is an acid, it is a nucleic acid. There are, as I told you, there are this phosphate which connects these phosphates are connecting the two the nucleotides. Okay. Now, so this O minus makes it an acid, actually this is OH, phosphoric acid derivative. So, now we start with, if we compare this part with phosphoric acid is nothing but P double bond O, OH, OH, OH. It is a tribasic acid. Out of these 3 OHs, which are the acidic hydrogens, one OH is forming an ester with the 5 prime OH, another OH is forming an ester with the 3 prime OH. So, this linkage is what is called phosphodiester linkage. So, if someone asks you that what is the linkage between the nucleotides, between the building blocks, it is the phosphodiester linkage. So, this is an acid because there one ionizable hydrogen is still there, and at the biological pH it will be O minus, and this will be definitely will be, there will be some water because there is water surrounding it in the biological medium. So, that will be that can be hydrated, but if it is put in a very anhydrous condition, then the DNA becomes more compact because not many of the water molecules are there. So, that is why A DNA has around 11 base pairs per complete turn, but B DNA, that is our normal DNA under normal condition, that has got 10 base pairs, 10 base pairs per complete turn. Okay. So, these are some basically some statistics about this DNA structure. Okay. Now, the next is that what happens. The next first thing is happens that DNA, yes, it is okay, DNA 2 strands are connected in antiparallel fashion by hydrogen bonds. Can we break these hydrogen bonds? Okay. Yes, you can break these hydrogen bonds, because after all these are not covalent bonds. Even covalent bonds can be broken if you raise the temperature to very high condition, molecules will dissociate, right. So, here also as you raise the temperature, then DNA, the DNA is a double helix. So, at a very narrow temperature range, what has been found that the DNA becomes separated, the two strands of DNA becomes separate, but that happens within a very narrow range. Why narrow range? It is not that as I raise the temperature from 30 to 35, 5% of the hydrogen bonds are broken, then going to 35 to 40, another 5%, not like that. Initially there is, you see, the way to follow this breakage of hydrogen bond is to study the absorbance at the lambda max of the DNA. See, DNA has bases which are aromatic in nature. So, they absorb at 260 nanometer, around 260 nanometer is their maximum absorption, lambda max. So, what you do, if you take the double stranded DNA and measure the lambda max at 260 nm, you start at temperature say 25 degrees centigrade. Now, you start heating the DNA. So, what you will see that the absorbance initially very slowly increases, not much, very slowly increases, and finally, at a certain narrow range of temperature, it just increases and then becomes stationary. So, this will be the absorbance increment curve. Remember, we are measuring the absorbance at different temperatures at 260 nanometer. Okay. Now, the question is, there may be a question that why it is such a within the narrow range of temperature, why all these are broken almost at the same time. This is what is called a cooperative effect, means somewhere if there is some breakage starts, immediately it the other hydrogen base pairs follow that. It is like if you have a stack of bricks, and if you push one brick which then push the next one. So, then all the bricks ultimately will fall from the upright position into the horizontal position, that you can do it. So, that is called cooperative effect, although you have only disturbed only one brick, but the whole brick system falls. So, here is also when you break certain number of hydrogen bonds, initially certain number of hydrogen bonds, the whole thing now collapses. Okay, it is a cooperative effect. So, this cooperative process makes it happens within a very narrow range of temperature. Okay. Now, this is a graph which is obtained by what you do, you plot the relative absorbance at 260 nanometer. Relative absorbance means some with relation to some absorbance. So, what you do, you make the first measure the absorbance at 25 degree and then you raise the temperature, say take 30 degree, what is the absorbance, then at 35, and then whatever absorbance values you are getting, you actually divide that by the absorbance at 25 and multiply it by 100. So, percentage, you can express it in percentage or you can express it in actual values, that is also possible. But what is important here is that the absorbance increases. See, not only the hydrogen bond is broken within a narrow range of temperature via a cooperative process, but the absorbance also increases and then becomes stationary. That happens because the DNA bases are initially stacked inside of the double helix. So, there will be less absorption of light at that time, and when the hydrogen bonds are broken, so the bases are now more exposed to the UV light. So, there will be more absorption, as simple as that. Okay. So, as you break it, the bases are now free to absorb, otherwise earlier they are all stacked together inside, congested in a narrow pocket inside the DNA. Okay. So, as it becomes open, it absorbs more. So, there is an increase of absorption. Now, in this curve, if you take the middle point of this rise, this sigmoid curve, this middle point, that means where I can say half of the hydrogen bonds, 50% of the hydrogen bonds are broken at the middle point. Okay. So, that is what is called the melting temperature of DNA. Melting temperature of DNA. So, when half of the hydrogen bonds, that means you can say 50%, see it is the definition is given here, Tm is defined as the temperature at which half of the maximum absorption increase is attained. That is one way of saying it, but you are not telling about the structure by giving this definition. You tell about the structure if you say that 50% of the hydrogen bonds are broken at the melting temperature. It is not melting of organic compounds, remember, it is not going from solid to liquid, it is actually the separation of the hydrogen bonds, and we are talking about percentage of hydrogen bonds that been broken. So, we have a number now 50%, the temperature at which 50% of the hydrogen bonds are broken is called the melting temperature of the DNA. Okay. Now, this melting temperature is very important, because if a species has a melting temperature of DNA say very low. So, what will happen as the environmental temperature increases? So, for that species the DNA will become single stranded, and if DNA becomes single stranded, all the subsequent processes like the flow of information which are called replication, transcription, all these processes will fall apart. DNA has to be double stranded in order to transmit all the information. So, nature has created this DNAs in such a way that the melting temperature is way above the environmental temperature for most of the species. Like most of the species it will be above 70 or above 80, but temperature will never reach like that. The maximum temperature what would be 50 degree in Rajasthan somewhere. So, but so the DNA is still intact as a single stranded there, and at the body temperature 37.2 there is no problem, 37 degree centigrade it is the melting temperature is such that it is so high that it remains in the double strand. To maintain a double strand is very important. Now, the reverse is also true. See, when you heat the double stranded DNA, so it becomes single stranded. Now, as if we think that I will cool the solution. So, if I cool the solution, what is going to happen? Now, these strands will slowly come together and try to find the complementarity in the other strand. So, at some point of time, then they will also join together. Okay. So, ultimately become double strand. So, as you cool, it will become a double strand. Okay. But the rate of strand separation and the rate of this annihilation, which is also called hybridization, that when two strands find the complementarity and become a double strand, that is what is called hybridization. But DNAs will hybridize. The rate of hybridization will obviously depend on the concentration that you are using, because after all it is the concentration, that how close are these molecules are, that will decide the rate, but the but 50% that rate of that, remember the point that you can again bring back the double helix by cooling the same solution. So, if you heat forms a single helix, single strand, and then as you cool it becomes the double strand again. Okay. This is also very important when we read the molecular biology. Now, the question is, what are the factors on which melting temperature depends? What are the factors on which Tm depends? Obviously, if you have a DNA where there is more percentage of G, that means there is more percentage of C, that means there are more percentage of these 3 hydrogen bonds that are between G and C. So, if you have more G content, that means you have more C content. So, that means your hydrogen bonds are more strong now. Now, the two strands are connected by a larger number of hydrogen bonds, because G is more, C is more. Okay. So, your Tm will increase, because it will require more energy now to separate the strands. So, the first thing that Tm depends on the percentage of GC content. Okay. And also this you should also remember that for DNA analysis you do not have to, suppose you want to know what is the percentage of G in a DNA, G, C, Ta. If you know the percentage of any one of the base, you can calculate the percentage of the other bases. Suppose in a DNA you have percentage of A is say 20%. So, that will give you the percentage of all the other 3, because if A is 20%, T is 20%, and so that will consume 40%. So, out of the rest 60, G is 30 and C is 30. So, you do not have to analyze all, you can analyze only one and get the percentage, and you, by the way, you can get the other ones. Okay. So, the Tm depends on the percentage of G and C. The other is Tm depends on the length of the DNA. The greater the length of the DNA, so the more number of hydrogen bonds you have, so that means you require more energy to separate that into the single strand. So, if it is a shorter DNA, the Tm will be less. If it is a larger DNA, Tm will be more. Okay. Then Tm, the Tm also depends on, suppose if you are taking determining Tm of a DNA in presence of a molecule which disrupts hydrogen bond, like urea. Urea is a famous molecule which disrupts hydrogen bonds in protein as well as in your DNA. So, it is a hydrogen bond disruptor. So, if you try to determine Tm in presence of a hydrogen bond disruptor, so your Tm will fall, because that hydrogen bond disruptor will assist in destroying the hydrogen bonds. Okay. So, things like urea, if you add, that will also lower the Tm, because it helps in breakage of the double bond, that hydrogen bond. There is another fourth point that is also important. If you take an ionic compound, suppose you are taking a DNA in a sodium chloride solution, okay, and then trying to determine the melting temperatures in sodium chloride. Now, you got a melting temperature of say 60 degree, then you increase the concentration of the sodium chloride, and the question is what will happen to the Tm. If you increase the concentration of sodium chloride, now what will happen, because of the presence of excess chloride ion, now the DNA will repel, the phosphate will repel these chloride ions, and so the two strands will be more intimately connected. They do not want to separate, because they are all surrounded by chloride ions. So, they want to push this DNA and make it more stronger, more strong, and if it does that, that means higher concentration of sodium chloride will higher the melting temperature of the DNA. So, these are the 4 factors. One is GC content, the other is the length of the DNA, the third is hydrogen bond disruptor, and the fourth is the presence of ionic compounds like sodium chloride. Okay. That on that your melting temperature depends. Okay. So, now the next thing, that when we started the protein chemistry or the peptide chemistry, what we know that there is a hierarchical structure in proteins, primary, secondary, tertiary and quaternary sometimes. In DNA, it is already double helix. Okay. So, more or less the 3-dimensional geometry is known. What is important here is to know the sequence of bases, that means how the bases are arranged one after another in a piece of DNA. That means, if a DNA has, the question is whether the DNA has A, then followed by G, followed by A, it is a random. So, there could be different types of sequences that are possible in a DNA, but these sequences are so important, because that will decide that what type of protein will ultimately be made. Okay. So, when we talk about protein sequencing, primary sequencing means when you want to know the covalent primary structure means where covalent bond is involved in the joining of the units. So, primary structure amino acid sequence. In DNA, structure analysis means this DNA base sequencing. That means, what is the sequence of bases, because other things remain same. Other things means the sugar is the same all the time, either in DNA it is deoxyribose, and the connecting bond is phosphodiester linkage. So, the difference between two sets of DNA is basically the sequence of bases that are there in the DNA. So, it's a challenge, but until this was solved, the DNA chemistry was difficult to take forward further, because what was needed is how to know the sequence of DNA, and as I said, this is important because that differentiates one DNA from the other DNA. Now, what is the first of all, what are the problems of doing DNA sequence? The problem is that remember in protein there is this technique called end-band degradation, which reacts with the N terminus and then breaks the first amino acid comes out, and the remaining peptide you would again isolate and do the end-band degradation. So, it is a repetitive process. Here the problem is the structure. If you want to know, suppose the DNA is having this type of sequence, and suppose this is the 5 prime end and this is the 3 prime end. If you want to know the structure of what is the first one, so you should develop a technique where only the first base is taken out, okay, and then your remaining part you isolate, and then you should develop a technique which must be different, because the second one is G. It should not be the problem is whether first of all, how different are A and G. Both are purine bases. So, it will be very hard to find a condition where only A will be cleaved, that means the A containing the sugar phosphate, that will be cleaved, and you will get a DNA which is only devoid of A, because A and G are very similar in their structure and in their there is slight difference in their reactivity, obviously, because one has got only NH2 6 amino purine, the other is there is a carbonyl at the 6th position, and there is another NH2 there, okay, NH2 there. Okay. So, the problem is how to find out very selective reagents which will cleave at either A or G or C or T. So, that was the challenge, how to do that. And the other thing is that you have a you start with a double stranded DNA, anti-parallel 5 prime 3 prime 3 prime 5 prime. So, initially you need to separate these strands, because you cannot do the sequence in both the strands at the same time in the same solution, that is not possible. But one thing is, you know that if you know the sequence of one strand, you can immediately write the sequence of the other strand, because you know that suppose if 5 prime to 3 prime is like this A, G, C, T, A, T, and I ask you to write the complementary strand, you can immediately write 3 prime T, C, G, A, T, A. So, that will be the other strand. So, you do not need to know the sequence of both the strands, just one strand you know. So, the initial thing that you must do is to isolate only one strand. That you can do by melting the DNA, and then before it can hybridize back, you separate these two strands. That is possible. You can separate these strands from a solution. So, the starting point is that you have a DNA which is a single strand. But before that, of course, there is some other important point which you have to consider, is that that when the DNAs are cut into, because we have to cut at selected points of the DNA, and then we have to try to locate these pieces. Right? Now, the question is DNA visible to your eyes? No, you cannot, it is not visible in eyes. Is protein visible in eyes? Yeah, there are some green fluorescence proteins, some fluorescence proteins which are visible if you shine light, but DNAs are not. Okay. So, like protein, you know, there is a dye which was called Kumasi blue. So, Kumasi blue goes and binds to the protein, and then you can wash out the remaining Kumasi blue, and wherever there is proteins, only blue bands you can see. Similarly, in DNA you have to have something which will show you the presence of the DNA at a particular location. Okay. So, that is so basically the principle is like this: the DNA is broken into pieces at precise points, and then these pieces are put in an electrophoresis gel. You know what is electrophoresis now? Electrophoresis is basically you put the material, and depending on the charge, if you apply voltage, then depending on the charge it moves. Now, DNA is always negatively charged because it is a phosphate, it is a phosphoric acid, remember, it is a phosphodiester, that means one of the ionizable weight is still there. Okay. So, it is an acid. So, in biological pH it is always negatively charged. So, the DNA will definitely move in this direction towards the anode. Okay. Now, if there are different DNA pieces of different sizes, so what will happen? You will have a distribution like this. The smaller size will move fastest, and the larger size will move slower. But again the question is, you will not be able to see the DNA here, because the DNAs are invisible. There you do it in a matrix. In DNA, in protein you do a polyacrylamide gel. In DNA there is another carbohydrate based material which is called agarose. So, it is called agarose gel in case of DNA, but the principle is same, that you apply the voltage and then the DNA being negatively charged. Here, unlike protein which has an isoelectric point, so there you have to take care of the isoelectric point, and depending on that it will move either to the cathode or the anode. In DNA that is not the problem, it will always move to the anode, the positive electrode. Okay. And depending on the size, the smaller size moves fastest and the larger size moves slower. Now, to identify where are the DNAs, what you do, you have to put some label in the DNA, so that suppose I put a label in the DNA, I put it a green, and then I break here. So, I have this green, suppose is when I shine light, I see the green, not green, the blue ones I can see. So, all these DNAs will have this blue once I shine light, but this is if it is a fluorescence reporter. So, I should have a tag or a reporter system which will allow me to visualize this DNA. There are different techniques that are available. Initially, it is only the radioactive technique that was used. That means, instead of fluorescence, you put a radioactive tag here. So, wherever there are DNAs, those areas will become radioactive because of the presence of the radioactive tag. And so, if you now take a photographic plate and put on the top of this, that photographic plate will be affected by only those zones from where the radiation is coming, because you have radioactive material in each of these DNAs. So, we will clear this. These are some of the fundamental principles by which you can do it by fluorescence, but fluorescence came much later. Initially, it was done by your radioactive phosphorus. And in the next session I will discuss that, but broadly speaking there are two methods which are parallelly discovered at the same time. One is called the Maxim-Gilbert method. This is a chemical method, and the other is called Sanger's method, Sanger's dideoxy method. Okay. And in the next session we will discuss these Maxim-Gilbert and Sanger. Thank you.